Okay, it's 11 a.m. Good morning, good afternoon, everyone. I'm Cherry Kubota. Welcome to Indoor Ag Science Cafe. August. My gosh, it's end of summer already. I hope you are doing good, and then I hope your summer was good. I really appreciate being together today again, monthly webinar for moving controlled environment agriculture technology forward. So just quickly, this opportunity originally started with a group funded by USDA we call OptimIA, and then now we expanded the scope, not just OptimIA focusing indoor vertical farms, but also greenhouses, high tunnels, so just much wider protected agriculture, indoor agriculture is our focus. And then we invite speakers from both academic community and then also industry sectors. And then academic speakers could be just a single, PI type of presentation or a group of academic groups to provide the project information, collaboration project information, or showcasing showcasing graduate students, postdocs. So that's variety of things to do, science-based information from academia. Industry speakers, basically you can introduce what you are doing in the company and what the challenges, what's the story, um, varieties of things, you can do in that type of speaking opportunity too. So, yeah, so I wanted to invite, or you can self nominate yourself if you are interested in speaking. So please take this opportunity once a month. We wanted to have speakers to share information, knowledge. Previous ones you can find in multiple websites, because this series of Indoor Ag Science Cafe is supported by 4 organizations or 4 groups, OptimIA, ADVANCEA, CEA HERB, and then United Greenhouse Growers Association. So if you go to either of the sites, you can find previous cafe recording. So just upcoming cafe. Today, we're going to have Dr. Mark Hoffmann from North Carolina State University. And then next month, we will have Dr. Jason Lanu. I may not be pronouncing his name right, but he is from Canadian Agriculture, Ag-Food Canada. So it's like a USDA equivalent organization, and talking about dynamic lighting. So that's really interesting. And then October, we have Utah State University faculty, Dr. Noah Langenfeld. He's going to talk about industry outlook, analysis based on USDA data. And then November, we have Casey Cole talking about some industry perspective. So with that, Dr. Mark Hoffman talking about advancing next generation strawberry propagation strategies. He's going to talk about achievements in the multi-state PIP propagation
I'll explain
indoor.
it.
Okay, PIP. I'm part of the project and not remember what is PIP standing. But anyway, PIP precision somewhere. But PIP CAP Strawberry Project. So this is the multi-year, multi-state effort. Thank you, Mark. Dr. Mark Hoffman, North Carolina State University. So I'm going to switch to Mark, and then he can take over.
All right, as you can see, I changed the title a little bit, but we're talking about the PIPCAP project, right? So first of all, thanks, Sherry, for giving me this opportunity. I'm very excited to talk to this group today. And I want to introduce myself a little bit, because I think most of you guys don't know who I am. So I just I'm a extension specialist, which means I'm a field guy, right? And I worked in citrus and strawberries and did my PhD in grapes, digging holes into the soil, and really just got into the controlled environment agriculture realm due to a very dear colleague of mine, Dr. Hernandez, who is also part of this project, right? And so it's all pretty new to me. I'm doing this now for like 6 years, and I am working at NC State for 9 years. I've been hired to serve the strawberry and the grape growers as extension specialist primarily, and I'm also an associate professor here at this university. And I saw a lot of overlap and opportunity in the CEA world, especially when it comes down to problems concerning the strawberry industry, not just in the United States, but globally, really. And that's one reason why I started leading this project. And I wanted to put this a little bit today in a little bit bigger perspective, but I'm going to share a lot of the project, a lot of the outcomes that we achieved in this project. So every time you see this little thing here popping up, PIPCAP, PIP stands for Precise Indoor Propagation, and CAP is the type of project that the USDA funded. And this project is now in its last month. We're doing this for 5 years. Sherry is a very invaluable part of this whole endeavor, and as are many others. And I want to say up front, I'm going to share a lot of outcomes and also some results. Not all of them are generated in my lab, and this at peak times, this multi-state project had more than 40 people working on several aspects of indoor propagation of strawberry plants. So I give credit where credit is due. And I also, as you can probably understand, will not be able to share every single result that we achieved in this project, because that would take an entire day. And we all don't have an entire day. We only have a few minutes, right? So it is my understanding also that not everybody is familiar with strawberries, so I want to give you a very quick overview about strawberry plants. This is something I show my students quite often in the class. Strawberries, as you all know, can produce fruit. They are coming from the sexual production organ, the flowers. But what we are focusing on today is the asexual reproduction, which is what the strawberry nursery industry uses to produce strawberry plants that are then annually sold to strawberry growers. So strawberries are grown in an annual system, and those plants are called daughter plants or sometimes tips. And then they grow on an above ground structure that is a stolon or runner, and typically every second internode has one of the strawberry daughter plants. That's not always true, but most of the time. A picture of a very young mother plant with stolons on it is shown here, and you can already see here that there are several types of stolons. There's a primary, secondary, sometimes there's a tertiary stolon, and all of those stolons can produce daughter plants, so it can become very quick, very complicated if you want to track the growth of a single mother plant over the lifespan of a propagation cycle. So I want to also introduce some of the plants that we're actually talking about in the industry. I think this is very important to understand that there are different plant types that are used in the industry on an annual basis and that are shipped out to growers on an annual basis in the United States. And the vast majority of plants that are used in the United States is on the left-hand side here, which is bare root plants. So those are plants that are grown in the field and then dug out from the field and then shipped to the grower, right? And there's a reason why people use this as a main source at the moment, but I would say a rough estimate between 90-95% of all plants that are used in the United States for strawberry production are those type of plants. One of the main reasons is because they're the cheapest plant material, right? And then there are other plants. Tray plants is what usually used or what greenhouse growers want to use. It's not very common in the United States. And then there's a 50 cc plug plant that is usually a 30-day or 28-day-old plant that mostly is used at the East Coast in some operations, right? So I also want to put down some of the challenges that controlled environment plant production has, because bare root plants are usually not the type of plant that are produced in any type of indoor environment. It's usually a rooted tip, which are both of the plant types on the right side, and that usually comes with a higher cost for the grower as well. The cost span was a lot higher than it is right now. When I started in 2017 here at NC State, the bare root plant sold for about 11 to 12 cents. Now we're at 30 to 40 cents per plant. So the span gets smaller, but it's still there. A fourth type of plant that we're talking about a lot today is the daughter plant or the tip. And again, there are specialized operations that do grow those daughter plants to then either produce their own rooted plants or to sell those daughter plants to specialized producers that produce rooted plants. And those plants are typically going for 20 to 25 cents per plant this season. So prices go up quite a bit on those commodities, right? So now, the United States is the second largest producer of strawberries in the world. The largest is China by a large margin. But when it comes down to productivity, so how many pounds of strawberries on average are produced per acre in the United States is by far the leading nation in the world. 46,000 pounds per acre on average are produced in the United States. The amount of acreage that's cultivated in strawberries is relatively small, with 70,000 acres. China has over 400,000 acres cultivated in strawberries. And that generates roughly $3.5 billion crop every year. So it's a really big industry, right? In total, we have the US retail strawberry market, which is by far the largest strawberry market in the United States, is a total of roughly $5 billion market. 3.5 again are somewhat produced in the United States, and then the rest comes from Mexico, right? And that is by far the majority of the strawberries that are sold in the United States are going through the retail shelf. There is a smaller market, which I call the niche markets, which is direct to consumer or greenhouse grown strawberries or local produce, which then usually are sold for higher prices than strawberries in the shelf, right? So if we focus on the retail market of the United States, we know that it's pretty centralized, and the main production regions are California and Mexico, and then Florida picks up about $300 million worth of strawberries in the winter, right? So also in the United States, what you see here is by far the main production system of strawberries currently, what we call annual hill plasticulture. It's an outdoor system, so there's very little indoor growing in the United States and on the strawberry market at the moment. The vast majority of strawberries, over 90% of strawberries produced in the United States, are produced in those systems that you can see here, like either the 2-row or the 4-row system, right? And those systems do rely heavily on soil fumigation, what you can see here, and 2 different types of soil fumigation. I'm not going to go into the specific, but that's still a big thing. And until early 2000s, especially in California, the California industry became extremely big due to the invention of methyl bromide and methyl bromide fumigation, which really helped the strawberry industry in California to get off the ground, really, in the latter half of the last century. And that today leads to California producing roughly, depending on the year, between 80 and 90% of all retail strawberries. They're not grown in methyl bromide fumigated ground anymore. At least the fruit are not. But they're grown typically in fumigated ground. Typically it's a chlorpicrin or methyl or like a metam sodium fumigation. And these are numbers from, I believe, 2024. Where California had about 42,000 acres of strawberries. And then if you see at this map, it's mostly in the southern coastal regions. So it's Watsonville, Salinas is what we call the central Californian region. And then Santa Maria is a big growing area. I believe at the moment, acreage-wise, the largest growing area. And then Oxnard and Ventura and then down in Orange County in San Diego. That's where the majority of strawberries are grown in California, and where the majority of retail strawberries that you can buy in the store, especially over summer, are coming from. Just to put it into perspective of volume, this is a typical 3-year average production curve just from California. And you can see here, first of all, California produces all year round. Several regions in California can produce all year round, especially the Santa Maria region has a very good climate and can produce in winter and in summer. But you can see here the bulk of the strawberries comes in between March and September, which really pushes the price of the strawberry down. In the retail shelf, sometimes a pound goes like for less than $2 to the consumer. And you can see here we're talking about almost 10 million trays, with a tray being roughly 15 to 18 pounds of strawberries. So we're talking massively high volumes of strawberries that are produced every year in California. Most of this bulk is produced in Santa Maria and the Watsonville area in summer, and then Oxnard and Ventura and a little bit of San Diego comes in the winter area, with San Diego picking earlier and then Oxnard and Ventura picking somewhere in this window here, right? So this is important to understand, because the strawberry nursery industry, which dominates the markets in the United States, has been tied with the strawberry production industry in California. So most of the strawberry nurseries and the large strawberry nurseries are all at the West Coast. Most of them are in California or Southern Oregon. And the retail market, so everything, all the growers that I showed you right now and all the strawberries that are produced are dominantly produced out of bare root plant material. So just like to understand what the current system is, right? So the reason that is because they're easy to ship, they're easy to plant, they're the cheapest plant material, and there's already capital investment into those nurseries already. Those nurseries have very tight relationships with the growers. They know when to produce plants and how many chilling hours they need to get, et cetera, et cetera. So it's a pretty tight relationship with capital investments into nurseries already made, right? So that's one of the reasons why this California nursery industry has become the largest nursery industry in the United States, because they serve the largest strawberry industry in the United States, which is California. And then they also serve a lot of Mexico, because a lot of Californian companies are growing strawberries in Mexico. So another reason is it is also very expensive to grow strawberries, not just in California. That is true for most strawberry growers across the country that are growing in soil systems, so in annual hill plasticulture systems. In California specifically, we're looking at almost $100,000 per acre to just grow. Growing cost. The majority of that are labor cost. But that puts growers typically on an average over a 5-year span on a 5%-ish profit margin if you want to grow strawberries. That is also true for smaller growers, at least to my knowledge. Most of the strawberry growers I know operate somewhere in this 5% profit margin range. So not very attractive, right? If you want to start growing strawberries, just be aware the profit is not very high. And it's also very challenging to change things to costlier, to add additional costs, because you're already looking at a very slim profit margin to begin with. Also, another thing that I wanted to share is that California alone probably needs 1 billion plants per year. That's about the amount of plants that they will consume every year, just to keep the operations going. And the nursery industry in California produces a lot more, because they produce also plants for Mexico partially, and they also produce plants for other regions in the world, really. So one thing that's really a challenge is the methyl bromide chlorpicrin fumigation in California. You can see here in California, and the specialized machines that gluing plastic to the soil are used to fumigate with methyl bromide. And that is a challenge for the strawberry nursery industry, because the strawberry nursery industry until today still relies heavily on methyl bromide fumigation. They have an use exemption that they have to renew every 2 years. And you can see here with the blue stars, those are areas where strawberry nurseries are in California. Some of the largest nurseries that have like several hundred, sometimes 800-900 acres of strawberry plants per year, are up here in the McDowell region, which is a very remote region of California, right? So estimates are that we're talking about 1.5 to 2 billion plants every year. Most of that is coming out of Northern California or somewhere here in Central California, where like smaller acreages are. And those nurseries has grown to really large firms that provide plants really to all regions in America and also internationally to Canada and other regions in the world. Just to give you like a small little overview how that looks like, we're looking at nurseries mostly up in the northern part of California. They mainly serve growing regions in the southern and central part of California, but also growing regions in Mexico. They serve growing and nurseries in Canada, as well as in regions at the East Coast, and also Florida, of course, is a big grower, and they grow a lot of plants for Florida growers as well. So this is a very complicated process, so I don't want to go too much into the details of what they do right now. I think it is important to understand to understand what the opportunities could be for controlled environment applications, right? And also to understand what the challenges are for those applications, right? So typically nurseries start out in tissue culture. They go through several steps of propagation in typically somewhat protected structures. They look like this, or look typically screen houses or something like that. And then they go into the open field where they already rely on fumigation, typically in increased blocks, and then into the production nurseries, which then have several hundred acres of plants planted. And then those plants, depending on when you need it and how many chill hours they need, and when your production cycle starts, can be high elevation nurseries, which are up north, or low elevation nurseries, which are more in the central part of North Carolina, of California. Just to show you like a picture, this is one field of a 900-acre nursery shortly before they start digging. So this was taken in July, and this you can see here's just like a sea of strawberry plants. They're all rooted in the field, and they have specialized processes to dig those strawberry plants, which I want to share with you. And this is the first thing I want to share from our project. We had our collaborator at Cal Poly was the Strawberry Center Director, Gerald Holmes, and he produced a video that I would love to share with you, at least a portion of that. So we tested this before. I'm going to try to do this now. There you go. So you see here, those are increased blocks, and then in those increased blocks, mother plants are planted, and those plants are being propagated and directly rooted in the field. You see, this is a very work-intensive process where those plants, bare root plants, are basically planted through specialized equipment. So this is all capital investment into those nurseries already, and then later in the season in those nurseries, you will have to remove weeds and flowers. Again, that is also very slow and very capital investment heavy work. You can see here a lot of the strawberry genetics, especially the newer strawberry genetics, do throw out both flowers and runners, and flowers do attract quite some diseases, especially if they become fruit, which is not wanted in the strawberry nursery. So there's a very diligent process of removing those flowers with people lying on those structures, right? So not pretty, but this is how it works, right? So this is the process that happens every time. And now we have to shop some diamonds, I guess, and talking to ChatGPT. I apologize for that. All right, perfect. And then I want to show you the process of harvesting daughter plants. So there's a three-way process usually, right? Like there's specialized equipment that harvests daughter plants that are previously mowed. So typically the daughter plants are mowed after they have the required amount of chilling hours. The chilling hours are important for daughter plants to provide vigor and also flowers in the fruiting fields later, and then those daughter plants are mowed and then harvested in those specific harvest machines in the field, and then put into like those big crates. And you can already see like one of the problems here is that if you have a diseased plant, then this definitely going to spread the disease quite a bit, right? So this is probably all I want to share on this one. And if you have any questions, please let me know. I'm happy to stop anytime. Yeah, so this is the process, and we were thinking part of the project that we was one of the questions was like, what are actually the main concerns of the industry, and how can we help, right? So, and that is work that has been done here at NC State by my colleague Daniel Trigiagel and his postdoc Zhu Shan. And one of the main concerns that the industry voiced over and over again was actually surprisingly not necessarily only the availability of methylbromide. That's certainly a concern for the industry, and a lot of industry folks were worried about losing methylbromide and then not having an alternative or an additional tool to use to make sure that disease is not spread through plant material. But a big concern was also the availability of labor. Those fields are extremely remote, and labor, like attracting labor to those areas, can be quite a challenge, and that has been voiced over and over by the industry as well. And of course, spreading disease through plant material, that has been quite a big issue throughout the time of the nurseries, especially starting with anthracnose or Colectoctium diseases in the past, and then of course providing enough chilling hours for those plants that need to be planted in planting windows. So jiggling the planting window and the chilling hour effect can be quite challenging too. So this nursery industry and is looking for additional tools, and then we were thinking that controlled environment can become one of those tools, right? And we were thinking about how could we integrate specific operations into either existing operations, like let's say in the screenhouse or an increase block, for example, or another avenue for controlled environment, another opportunity for controlled environment, was the development of completely new infrastructure with direct grower-facing entities closer to the growing regions, right? So those are kind of like the two main opportunities that we think where we think controlled environment and indoor strawberry propagation can be an additional tool helping the industry. So I want to make sure, if we go back to this slide, before I forget this, I want to make sure that especially the first three things, the availability of methylbromide and the availability of labor and disease spread and chilling hours, right? Like, so as long as nothing is moving on those parts, so as long there is methylbromide still available, as long as you can get enough labor in those remote regions, and as long as you can somehow provide the chilling hours that are needed for the strawberry growing regions, as long as nothing moves in this area, there's a very low likelihood of major capital investment into other structures, right? But if one of those things falls away, let's say there's no methylbromide anymore, or labor goes away, or you can't just not get the chilling hours anymore that you need, then that might trigger a investment into other capital structures or in other tools, right? So I also want to mention that there's another nursery industry, a smaller nursery industry that really is much more diverse, that produces rooted plants, that produces in hanging gutters, like you can see here, all like tips and fields, what you can see here as well. That nursery industry is mostly located at the East Coast, including in Canada, and doesn't serve the California industry, but it does serve a lot of growers at the East Coast and in central United States. That industry is a lot smaller acreage-wise and also output-wise, and has relied in the past quite heavily on stock material that came out of those large California nurseries. And just recently, there's a shift for those nurseries, or for most of those nurseries, to go into tissue culture rather than relying on stock material of California nurseries. But that's a relatively recent development. For most nurseries, there were just a handful that already started with tissue culture at the East Coast. Most of the nurseries at the East Coast relied on stock material from the West Coast. Yeah, those serve mostly niche markets, which I would say are mostly local produce, greenhouse, strawberry markets, and direct to consumer. And they usually achieve higher prices than in the retail space, more or less. They still compete with the retail space, but they can ask for higher prices due to several properties that they have, right? So in our PipCap project, we wanted to make plant propagation more efficient. We asked the question, how can we make this more efficient, and how can we make the main product more attractive, right? So those were the main two questions that we had in our PipCap project. We started this in 2022, really. We got the money in 2021 until everything was set up. It was January 2022. And we aimed to develop knowledge protocols to enhance indoor strawberry propagation. That was our main mission, right? And there was a big project with a lot of collaborators. As I said at the beginning, at the peak of this project, more than 40 people were working on, including graduate students and staff, were working on several aspects of indoor propagation of strawberries. And what we found out very quick was that we don't know anything about strawberries, right? So, for example, and these are some questions that we know, we had no idea how the nursery industry really works. It was a very, there's just a few firms. Every nursery industry does it a little bit different, and we didn't really have a great overview about the nursery industry. We didn't know how different cultivars react to different light treatments, for example. We didn't even know if the plants that we create are they better than what the industry standard is, right? Can we make them less costly? What are the differences in daughter plant production between cultivars? We didn't know any of this. And do we have a genetic basis for differences that we see in mother plant production, right? Like, so all of that and a lot of more questions we had that we couldn't answer, right? And so I want to give you some ideas what we did. And one thing that we really needed to know is what are the differences in propagation traits among cultivars. So we sat together with industry in 2021 already, and decided on several cultivars that we want to evaluate. And we had in total 13 cultivars that we evaluated. And then we put in the last 2.5 years of the project, we put those plants, we grew them from tissue culture, we rooted them in a chamber, right? And then we put them into a greenhouse hanging gutter system that was under artificial lighting to really evaluate how those different plants are propagating, right? And that is a project, again, I like to always highlight who is responsible for this. So this is a project that really started out with a graduate student in Ricardo's lab, Sam, who really gave us the ideas how we can even track the runnering of strawberry plants, because as you could see at the very beginning, it can become very complicated very quick. And then my team here in my lab really made the system more workable and visualized the system a little bit, and also did the hard work of actually tracking every single daughter plant over 13 cultivars over 2.5 years in this system. So I want to show you one of the other outputs that we have. So this is something that at the moment is not online yet, but we had 4 batches. We're at the moment visualizing batch A and batch B. There are 2 more batches coming. And we're going to put this onto our homepage as a Strawberry Trait Explorer. You can see here between the different stolons. You can look at the different stolons. We even had quaternary stolons for some of the cultivars. And then, of course, primary, secondary, and tertiary daughter plants. And you can go, for example, at total daughter plants on alternate nodes, which are the most daughter plants. You can look at the cultivars. So let's say we want to compare Albion to Sensation, for example, right? Then we're going to do this. And then you see this graph here. that can explore the number of daughter plants over time produced by albion sensation. So you can play around with this. This is going to be online, hopefully by end of the month. At the moment, this is just for us to play around. There's some bugs in it that we need to figure out. But also pretty cool thing, Sherry, you're gonna appreciate this, is our plant animation part. So this is based on our mother plant experiences that we had with certain cultivars. And let's say we can look at a very good runner producer, which is Radiance, and then you can play the production of daughter plants and runners over time at this specific mother plant in our simulator, right? And then we can look at different mother plants, for example, this one here. And then you can see here, those are tertiary, this is a secondary stolon, this is a tertiary stolon. Those are daughter plants on the tertiary stolon already. So you can see here how those stolons produce, right? And then you can compare this, let's say, for example, with like a cultivar that doesn't produce that many runners. And you can see here, again, in our play. Yeah, again, so there's some bugs in it that we still have to figure out, but this will be available pretty soon because we're already running the tests on it. All right, let me go back to the PowerPoint. All right, we also want to look at, so there are clear differences in runner development, stolon development, and daughter plant development. So we wanted to see if we find any gene variants that can dictate runner production. And that has been a team collaboration between my team here at NC State and the genetics team at the University of Maryland. And we focused on one specific gene in this gene. As you can see here, typically flowering is usually get a bigger flower response under cool temperatures and under short days, because cool temperatures and short days inhibit TFL1, which then in turn promotes FT3, which is a flowering gene, right? So under long days and warm temperatures, that typically promotes TFL1, which is inhibitory to flowering, right? But and on the runnering part, it's exactly the other way around. So gibberellic acid basically inhibits DELA, which then inhibits, which DELA typically inhibits runnering, so that would promote runnering. So the more gibberellic acid is produced, typically you would see a bigger runner response, right? So we want to focus on that specific gene, GA20OX4, that actually codes for, this is one of the gibberellic acid coding genes, and we want to see if we find cultivars with a big runner response cultivars with a low runner response that might have mutations on that specific gene in one of the 4 subgenomes that we have in the strawberry plant, which is an octoploid. So to do this, we developed a quite comprehensive genome database, and I'm going to show you that in a minute. That genome database is already online, and we looked at different subgenomes, and then in subgenome C of Monterey and Fronteras, we actually found a 1 base pair insertion on Fronteras versus Monterey, which didn't show any mutation on that specific gene. So this 1 base pair insertion do render the gene unuseful, so which means you wouldn't see any gibberellic acid production coming out of that specific subgenome. And we also found that if we looked at Fronteras and Monterey compared in a control environment, we saw that Monterey produced many more daughter plants in a controlled environment, almost 200 daughter plants in a controlled environment over 10 weeks compared to Fronteras. So now I want to also show you the genome browser as one of the outcomes of our product. So this is I'm not a geneticist, so I only want to show you what we can do. We have a comprehensive search where you can multidimensionally search and explore several, for example, glutamate receptors, and then it's searching through the database. It's a very comprehensive database. As I said, we looked at 13 cultivars, we Illumina sequenced them, with a 40x depth for each of the alleles, and then you can look at what you find in this system. We also can look for gene descriptions, and we can look for specific gene names, right? And then of course we can look at the entire genome of cultivars, and then also of Arabidopsis homologs if you in our cultivar. So if you click at glutamate receptor 2.09, for example, we're loading the gene data. And then you can open your track selector and can look up the genes for example, Monterey and Fronteras. And then you're gonna see the inserts and deletions that are marked red and blue here on certain subgenomes on Monterey and Fronteras. So this genome database is already online. All right. Okay, so, and then of course, one big question is how can we optimize growing conditions across those three stages, mother plants, daughter plants, and then also how does this translate into the field, right? So that was one of the big questions we had as well. And we're going to focus a little bit on the mother plant part. And I want to say again up front, this was a pretty big objective of the project, and was done. Ricardo Hernandez was a leading PI there, and then Brian Jackson looked at root environmental responses, and then Jennifer Bold from the USDA in Ohio looked at nitrogen and salt responses of different genetics, right? So I had nothing to do with this, and everything I'm going to show now has nothing to do with me or my lab, and I'm not going to show you too much of this because as I said, this should be really up to Ricardo, Brian, and Jennifer to present in more detail. A majority of the work on the mother plant impact of different control environment treatments on mother plant performance was done in the Ricardo Hernandez lab, and I'm going to just show you all the lab members here. Really important is Sam and Moen. So Sam was a master student and Moen was a PhD student in the Ricardo Hernandez lab. Both of them did tremendous work here at NC State. And again, I'm not going to highlight all the work they did because I really want to leave that up to Ricardo. I'm just going to give you the outcome of this. Based on their calculations, in a number of daughter plants per square meter could increase in the controlled environment 16 times compared to what we find in the open field, right? So compared with what we find in the open field nurseries in terms of daughter plants per square meter. So there are some caveats to this. First of all, the open field daughter plant are estimates. There is no counting, right? Those are estimates that we got from the industry, so they're quite reliable, but there's no counting on how many daughter plants we have per square meter on this number. And then secondly, we're talking in the CEA system, we're talking tips. We're talking this structure here, right? Like the structure that we use to develop rooted tips. And in an open field nursery, this number is coming from a bare root nursery, which we're talking about the final product that can be sent directly to the grower. There is another step between here and the final product that you want to sell to the grower. Well, this is the final product that can be sold to the grower. So that's the difference. But this is quite a stunning number, even though we compare two different products with each other. And I want to highlight what happened in the Ricardo Hernandez lab without going into the research details. But they looked at photoperiod, at spectral light intensity, did quite some work on light intensity. Sam looked at CO2 levels, and Moen looked at inter-canopy lighting and plant density. So that is something probably from other Strawberry Cafe when Ricardo wants to show those results in more detail. I leave it at that, but I want to show you our homepage, which is where you do find a lot of and I'm going to show you where that actually is. If you go to our homepage, you find a lot of the stuff, especially into our knowledge center. You find project news and publications. And then you also find presentations. So if you look at presentations from our last annual meeting, all those presentations are online. They're very detailed, and you can find all and a lot more information on what I just talked about here in those presentations, right? So please go to our homepage and make use of this. We have quite a lot of also our publications are online here as well, and then of course our dissertations and theses are also here to read. So there's quite a good homepage to visit, right? And then of course you can also get access to our genomic database through the same homepage by clicking Access Database, right? So so you find a lot of the details of Ricardo's work on the homepage. I want to talk a little bit more about Jennifer's work because I understand it better. That's why, and I also think Jennifer is part of this of today's crew. So if I do something, get something wrong, she can just intervene and say something, right? So, but I think it was quite interesting. Jennifer, I also want to introduce the team. I don't know all of those people. The only person I know is Aaron, who was a postdoc at the at the PIPCAP project. And I think the main takeaway of this work was that we know that EC and nitrogen fertilizer form can play a large role in the amount to optimize strawberry daughter plant production, right? So that was kind of the takeaway. And I want to show you some of the graphs from Jennifer's work. Those are also published in 2025. And again, the responses are quite cultivar dependent. And you can see here that Fronteras and Monterey do really have a peak in nitrogen form somewhere between 60 and 80% of NO3 versus NH4, while Albion more showed a more or less linear response to the amount of NO3 in terms of output, which is number of daughter plants per plant, right? So also shows you here what we saw in the other project, in the other graph, that Fronteras is not the best runner on the planet, and Monterey produces a lot more runners, right? Also with EC, we found quite some peaks between Monterey and Albion, very similar. Albion seemed to have like a little bit lower peak, somewhere between 1.5 and 2.0, while Monterey can get a little bit higher, somewhere around 2.5 as the main peak before production goes down again. So there was very critical work. Fertilizer and nitrogen concentration didn't seem to have that much of an impact compared to EC and nitrogen form on daughter plant number. So again, this was Albion, Fronteras, and Monterey. And you saw like a little hump, but it's not, doesn't seem to be as impactful as EC and nitrogen form. We also looked at daughter plants, and we looked at a lot of different things at daughter plants, from storage and improvement of flowering and runnering. And I also want to highlight a little bit of that. This was a project that Sherry did lead, and we had several people working on this. Selena Gomez from Purdue University looked a lot into storage. Edward Derner from Rutgers looked at light, and Sherry and we looked at temperature and chilling requirements and chilling impact of strawberry plants. And I do also want to highlight a little bit of that. These are the two PhD students. This is Pooja, that's Sherry's PhD student, and this is Michael. This was my PhD student, and both of us looked at the impact of chill hours on the development of rooted strawberry plants, just at different developmental stages, right? Where Pooja looked at a lot higher chilling hours and how a lot more chilling hours, and looked how it impacted runnering, while we looked at lower amount of chilling hours and wanted to see in an earlier stage of plant development if we can affect flowering, right? So I'm gonna talk a little bit about that. And one of the outcomes here was that supplemental chilling, which is often done in nurseries to provide additional chilling hours in the cooler, in the dark, we didn't find any effect on meristem fade. We did a lot of flower mapping over the last 3 years. But what we found is when we simulated day-night growth cycles where we get chilling hours at night in the growth chamber, we found that at the same age plants, under the same amount of chilling hours, we actually found some impact on meristem fade based on those, right? So I want to show you this in graphs a little bit. This is what we evaluated. And you can see here what like most important is the runners and trusses, and we found here that we didn't give any chilling hours all the way to 600 chilling hours, and we didn't find any impact of any chill hours that was significant on either the development of runners or trusses on any of those plants. And this is for Monterey, but we did this on 4 different cultivars. We did this with Monterey, Albion, Sensation, and Chandler, and we saw the same picture basically across all of those. That is not true if we do simulate natural conditions. So if we do give day-night cycles where we do dip into chilling hours, 4 degrees Celsius, overnight, over the dark period, and then gave growth conditions over day, that impacted quite a bit, especially truss development and runner development. And that's especially true for larger size tips, right? So you can see that here, that especially for Monterey, we found quite a significant impact. If the tip size was large enough that they carry enough meristem to actually have an impact, right? So this was done on plants that were 4 weeks rooted and then put into a grow chamber. Yeah, and then we looked at field performance of plants as well. And that we want to know if our control environment produced plants provide similar or better yields than commercial standards. We did 2 years of field trials in different regions, of course, in California and Florida, also here in North Carolina. And we gave chilling hours and non-chilling hours to plug plants and then compared them to bare root plants in those areas where bare root plants are the predominant commercial standard. And then, of course, to commercial plugs where they are standard here in North Carolina. And there are a couple of takeaways from this. Field trials are always more complicated than they seem. But one thing that's pretty clear is if you get a year where you do have a diseased plant material from a commercial grower, which we had in 2024, the plant material that comes out of the chamber performs significantly better than the diseased plant material. This was happened in 2024. You can see here the commercial plug plant was infested with neopistolopsiosis, a disease that can be spread through plant material, through open field nurseries. And you can see here that even the non-chilled cultivar was performing better than the chilled, than the commercial variant. So in years where you don't have diseased material, that picture looks a little bit different. Again, this is a different season, very different weather pattern. That's why we had like one big peak that season, right? But you can see here that all the material kind of performed the same way compared to last year where we had diseased material and therefore less performance of the commercial varieties. So in areas where we do need chilling hours, which is Central California and Southern California, which don't provide enough chilling hours over the winter to really provide a significant amount, in those areas, we always saw that the chilled plug variant did perform significantly better than the no chill, and of course somewhere on the same level than the bare root plant that was planted simultaneously at the same time, right? And they got more or less the same amount of chilling hours. That is also true for Southern California, with some exception. What we often see at early planting stages is that we do see a peak of plug plants early on, and then the bare root plant comes in later in the season. That is something that might be wanted or not wanted, really depending on what the grower's main window of sales are, right? But what we did see in our chilled plug variant is that we saw a peak of the chilled plants early in the season, while the bare roots then came on later in the season, which is pretty typical and described before for this specific region, Southern California. And then in regions where we do not need chill hours or where only low chill hours required, which is Florida for the most part, we didn't see a big difference between our chilled and non-chilled plug plants, and the bare root material seemed to work a little bit better, at least in the field trials that we performed on Florida 127, which is Sweet Sensation, and also we did the same thing on Brilliance. So What we learned was that artificial chilling is extremely cultivar dependent and may be required for open field applications of controlled environment rooted strawberry plants, depending on where they are grown. And what we also learned is that day-night chilling with night chilling and day temperature, growth temperature affects young plants differently compared to supplemental chilling, which is pretty common in the industry. And again, you find a lot more information on our knowledge center. I unfortunately only have an hour to share with this, and I want to also share before I end this presentation that we're working on a guide to propagate the strawberries. This guide is already in review, so I already went through one round of edits. I have it on my table right now for a second round of edits, and we hope to get this guide out as an extension publication September 26th. It's pretty comprehensive, has over 100 pages, and that hopefully will be done later this year. So now these are all the people. Way too many to all name by name, but those are all the PIs that helped getting this project done, and Lisette Vigil, our project manager. And those are all the postdocs and PhD students and staff that worked on this project over the lifetime of this project. It's a lot of people, and I really want to thank every single one of them, because wouldn't be possible without them. And I call this challenges and opportunities. So I think one of the main challenges for indoor production is the capital investment, of course, and also the current production systems in the United States does seem to favor bare root plants, and the price point and the superiority of plant material coming out of CA productions is also a challenge, right? But there's also opportunities, especially because a system like this can serve to propagate niche cultivars and niche markets. We do have a lot more cultivars on the market today than we had even 10 years ago. And it's a challenge for large nurseries to propagate those cultivars. And that could be a tool where indoor propagation can serve today. And then in the future, as I said, as soon as regulations change, climate or labor will change, that can be a pressure point where more capital investment goes into new tools. Right, so we're working on a second iteration of this project. Currently, we applied to SCRI funding recently, and the focus of this is on plant health, energy efficiency, cost, and automation. Again, a lot of collaborators on it, and this is my son, 6 months old. That's why I'm tired and get gray hair, but he's also a lot of fun. And thank you very much, and I'll take some questions.
Thank you. Thank you so much. I really appreciate.